Programmed cell death, or apoptosis, is a necessary process in all living organisms. It's the process by which cells are replaced when they become diseased or damaged. Within the membrane of a cell, excess protons, positively charged atoms, are transferred through a regulating pump. The rate of this process is thought to influence whether a cell should live or die, according to the needs of the body, by indirectly regulating the expression of two proteins, FLIP and XIAP. These proteins prevent executioner enzymes called caspases from telling a healthy cell to die. When the cell encounters a death signal, the caspases are no longer regulated by FLIP and XIAP, creating a series of events that lead to apoptosis or programmed cell death in a cell which has reached the end of its useful life. Cancer cells do not effectively respond to these normal death signals, resulting in a tumor. Proper regulation of caspase inhibitors is critical to the function of programmed cell death. Phenoxodiol is an experimental anti-cancer drug that appears to have no effect on healthy cells but is highly selective in targeting cancer cells. The ability of phenoxodiol to act against cancer cells is thought to be due to its disruption of several key targets specific to cancer cells. One of these targets is this regulating proton pump. In cancer cells, the proton pump operates more rapidly, indirectly causing more inhibitor proteins such as FLIP and XIAP and preventing death signals from activating the caspases. Overexpression of these inhibitors explains resistance to many chemotherapies that work through the death signaling pathway. Phenoxodiol appears to be a highly selective compound targeting the overactive proton pump in cancer cells. Inhibition of the cancer cell proton pump causes accumulation of protons within the cell. As a consequence, the membrane signal transduction cascade is disrupted, thereby blocking the production of FLIP and XIAP proteins. The death signaling pathway can now respond to the signals delivered by chemotherapy, activating the caspases, which in turn initiate programmed cancer cell death. Thus, by exposing tumors to phenoxodiol, cancer cells appear to become more responsive to standard chemotherapy. In laboratory studies using human cell lines and animal models, phenoxodiol has been shown to be active in cells representing a broad range of cancers, but has no effect on healthy cells, explaining its safety profile.